in this section 2, where we started with membrane physiology of nerve and muscle, we designated to have 3 lectures. So, we are done with the first lecture, where we talked about the lipids which are present in the membrane and the classification of the lipids and few other details. Today, what we will do in this part, second lecture of this section, we will be talking a little bit about the structure of the membrane and the techniques which are being used. So, before I move on to this lecture, there is a small carryover which I missed out in the previous lecture. We talked about within the membrane, you have the presence of the glycolipids and the phospholipids. There is one more component which is present in the membrane and which has a very profound role to play, that is called cholesterol. So, now I will just add that and then I will start this today's section's lecture. Okay. What exactly is the cholesterol among the lipids? We talked about the phospholipids and the glycolipids. Now, this is the third moiety in that structure. This is a very profoundly important molecule in biology and evolution. This is how the structure of cholesterol looks like. It is a fairly big structure with the X. I will mention what X is, and you have one second. You have one CH3 attached here. You have another CH3 attached here, and you have a OH hydroxyl group attached here, and this X equals to your CH3 H, and this is where the X is actually connected, CH2 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 CH CH3 and CH3. So, this is the structure of the cholesterol, and in this lecture we will come to see where this cholesterol is playing a very important role in the fluid nature of the membrane and what are its other physiological function. As I will work through the course, we will talk about it, where the cholesterol plays a major role. With this small backlog which I missed upon, I will now enumerate the topics which I will be taking up in this lecture. There are 12 small topics which I will be taking up in this lecture under the heading of membrane structure and dynamics. So, we have already covered around 10 topics in this structure and dynamics. And the topics which I am going to cover today in this section, this includes lipid bilayer. First topic, lipid bilayer and techniques to study lipid bilayer. Okay. This is the first topic I am going to, techniques to study lipid bilayer, just putting it as BL as a shortcut. Then we will be talking about the permeability. This is the second topic I will be touching in this lecture, permeability of lipid bilayer. Then I will be talking about the flow of ions through lipid bilayer and how this could be detected. The fourth topic what I will be covering will be the membrane protein. The next topic I will be covering is the experiments to prove. If you remember, in the last lecture I told you I will be talking about the experiments to prove the mobility of proteins and lipids in the membrane. Next topic I will be talking about the fluid mosaic model of the membrane. Then we will be talking about the asymmetric nature, slightly more detail, asymmetric nature of the membrane. Followed by that, we will be talking about factors controlling membrane fluidity, and this is where we will be talking about the role of cholesterol, membrane fluidity. Then we will be talking about presence of carbohydrate in the membrane, and last we will be talking about the use of detergents in studying membrane proteins. Membrane proteins. So, these are the topics which I am going to pick up in this section. So, having said this, let us talk about the lipid bilayer. So, in the last class I told you that lipid bilayer, the bilayer formation is directly related to the presence of phospholipids and the glycolipids, and now I have added the cholesterol. So, now what are the different techniques which could be used to in order to understand this? So, under this heading we will be talking about, let us draw the lipid bilayer. So, this is how it looks like. Remember when we drew it like this. So, this is one side of the lipids, this is the other side of the lipids, and here you have the hydrophobic chain. So, is there a way how we can recreate this lipid bilayer in the lab? So, this is there are two different ways how you can do it. So, we will be talking about one of the techniques first, which will help you. So, that is basically fall under the preparation of lipid vesicles in order to study the lipid bilayer. So, how it is being done? So, preparation of lipid vesicles. Yes, okay. So, what is generally done in the labs is that you take a beaker like this. Okay. In the beaker you have a layer, you make a layer of phospholipid. Just put the phospholipid, you can see it in green. This is the phospholipids, PL. Okay. On top of that, you add water likewise, and you can use any kind of in this case I am adding, these are small amino acid called glycine. Okay. And this is water. What you do with this, you sonicate this. Sonicate means you agitate it at a very, very high sound frequency, sonication. So, once you sonicate it, so let us follow it up. Following sonication, what you see, it forms something like this. Let me just check what is the color code I used. Fine. Okay. So, post sonication, what you see these kind of structures which are formed, and this is the water all over the place outside, and within that you see the trapped molecules of glycine, and there are some glycine which are present outside. So, from here what you do a simple technique called gel filtration, and following gel filtration what you obtain are pure lipid vesicles. you get rid of all the glycine from the suspension, and there are glycines which are trapped inside the lipid vesicles. Here you have the water. This is how you create the lipid bilayer. This is one such techniques of creating pure lipid vesicles. And you can use the kind of whichsoever phospholipids you can use instead of phospholipids, some mixture of glycolipids, you can use cholesterol, you can vary the concentration of cholesterol, you can do several things in this situation, okay, which will help you to create different kind of vesicles. There is another technique which is slightly more advanced technique, that is called black lipid membrane technique of studying lipid bilayer. Black lipid membrane. In short, sometimes it is also referred to as BLM technique, black lipid membrane apparatus. So, what really black lipid membrane apparatus is? So, just imagine you have two beakers like this. You have a jar where there is a small partition, fine, and within that partition you have a small hole like this. Sorry, let me redraw it like this. Okay. Now, what you do, so you see this a small hole out here. Let me mark this hole for your understanding. So, this is where the hole is. Okay. And what you do, you take a paintbrush. All of you have seen a paintbrush. You take a paintbrush like this and dip it in. This is the paintbrush, and the dimension of this hole is approximately 1 millimeter. Okay. You take the paintbrush and you have the lipid solution here. It could be a phospholipid PL, it could be a glycolipid, it could be cholesterol, whatsoever. And then you take the paintbrush out here, just the way you do it like this. You rub the paintbrush out along that hole. So, once you do so, what will happen is this. If I redraw this, and here you have the gap, it forms a lipid bilayer here. So, something like this, what will you see? If I observed it under the microscope, it will look like this. On either side, it will look like this. So, in other words, you formed a membrane across that hole, something like this. This is what is going to form here. So, if I redraw it on top of it, it will be like this. Just for simplicity's sake, I am following a color code, but it could be all mixed up, something like this. Now, what you do is you fill this side with water and you fill the other side with water, and you add different kind of solutes into this. Okay. Say example, you add sodium, potassium, calcium, you do the same thing at different concentration, calcium plus sodium plus potassium plus at different concentration. You can have sodium, potassium higher on one side, potassium lower on another side, you can have sodium higher on one side, potassium on the other side. And then what you can test is actually you can connect it with two electrodes. You have electrode like this and you have another electrode which is like this, and these electrodes are connected to some kind of voltmeter, okay, which will measure the voltage, or you could kind of some other gadget which could measure the voltage. So, any kind of mobility of the, go to the next slide, so that it is not crowded for you to understand. So, here you have the bilayer already formed. Okay, and here is the electrode on one side, here is the electrode on the other side, and this is all connected to some kind of measuring device. So, this is the electrode, this is the electrode, and this is that hole where the lipid bilayer is formed, and here you have the fluid and with all the different ions. So, any kind of movement of ions across this could be recorded by this voltage sensor or any kind of current devices which are present there. So, this is how the black lipid membrane is so very useful in understanding the movement of any kind of ions or any kind of molecules across the membrane. Okay. So, this is one of the techniques which are being used. From here we will move on to the permeability of lipid bilayer. This is very important. This is the second topic of this section. Permeability of lipid bilayer. So, one of the fundamental thing about lipid bilayer is that these are highly impermeable to ions and most polar molecules. The molecules which are having some form of charge out there or some form of ion like sodium, potassium, they are exceptionally impermeable. They cannot really directly pass through the membrane. In other words, what I mean is this: say for example, you have a lipid bilayer like this, and something like this, and you have this it is very tough for these kind of ions or some kind of polar molecules, some kind of say polar amino acids which we have already talked. It is most unlikely that it can pass through. This will block the movement of these polar ions from either side, whichever way you try it, it is going to block the entry of the polar molecules. So, polar molecules or the ions, polar molecules and ions are very, very impermeable to the lipid bilayer. And if I kind of draw a scale of about the permeability of different component through the lipid bilayer in terms of the increased permeability, if I have something a scale like this, increase permeability, unit will be permeability with centimeter per second, that is it. Then if you look at it 10 to the power minus 14, just putting the values 10 to the power minus 12, 10 to the power minus 10. Likewise, if I move to 10 to the power minus 2, 10 to the power minus 4, 10 to the power minus 6, 10 to the power minus 8, 10 to the power, okay, I can just rub this is a scale, okay, 10 to the power minus 8. If this is the permeability values so you will see the most permeable will be water. Then you have something in around 10 to the power 4, you will have something a molecule called indole, which is very similar to another molecule which will come very soon. Then around 10 to the power 6, you have urea, which is more permeable, urea or glycerol. Then around 10 to the power 8, you have tryptophan. Tryptophan is one of the amino acids, and around 10 to the power 8 is glucose. Then comes around 10 to the power 10 is chloride ions, 10 to the power 12 is potassium ions, and near about is the sodium ions, which is very least permeable into the game. Now, if you look at this figure, just it raises a very simple question: how come this tryptophan, which has a very similar structure as compared to indole, very similar structure, how come indole is more permeable as compared to tryptophan? So, in other words, the question is why indole is more permeable through lipid bilayer as compared to tryptophan. This is one question I will leave for people to think over it. what is the difference between the two structure which makes tryptophan less permeable to lipid bilayer as compared to indole, though their structure is very, very similar. Okay. And your answer lies in some of the polar nature of it, as I have mentioned in this small section, that polar molecules or the charged ions or the ions are very less permeable across the lipid bilayer. So, now the question arises, a very serious question now arises, what makes an ion or polar molecules to pass through? There must be some way, because as we have already seen the previous scale, that most of these are not that very permeable, and this is where comes the role of proteins or the membrane protein. This is where comes the role of ion channels, ion channel proteins, which is a very, very significant area of research. There is a huge amount of budget which has been allocated by National Institute of Health in the United States and several other countries to understand the structure and function of ion channels, and we will talk more as we will move in through the course. Here comes the role of pumps. So, these are those proteins which are embedded within the lipid bilayer, which allows the flow of polar molecules, ions and many other molecules which directly cannot pass through lipid bilayer, or the permeability is very low, and with that permeability no physiological function can take place. Okay. So, now we will, in order to understand this, we will talk, before we get into any kind of membrane protein, which are very, very complex structure, and very little structural information about it is available currently. Very few are really known in at a very low resolution crystallographic structure. We will talk about some simple molecules which have the ability to form channels or pores along the membrane. So, this section falls under transport antibiotics as carriers or channels. Okay. So, now this is what we are going to deal with. Transport antibiotics are carriers or channels. Okay. So, talking about this, let us move on to the next slide. Let us redraw the membrane again. These are the polar head groups in red, and another set of polar head groups which are facing inside the cell like this. Okay. And here is the hydrophobic tail. I keep on repeating it so that it kind of engraves into your thinking and understanding. So, I am kind of continuously repeating all these things for you guys. So, a membrane protein or anything which has to form a passage has to stand like this. It has to form something like this, a structure like this, which has maybe an inner core like this, through which any of the molecules can pass through, something like this. Okay. This is the molecule which is passing through. Okay. So, this is outside and this is inside. Okay. So, what are those nature of these kind of molecules? This is where we are going to talk about some of the smaller ones, simplest one, before we get into the one which are forming ion channels and everything. Okay. So, one such example is carrier transport antibiotics are something like this. This is one such carrier transport antibiotics which has a hydrocarbon periphery, and within the hydrocarbon periphery it forms, it has something like this. So, this is called carrier transport antibiotics. I am just putting a short form of antibiotics as AB, so that you people will understand. These are the oxygen molecules, and in the center you have a potassium, and potassium ion form coordination complex with 6 oxygen there, and this is the zone which is hydrocarbon or hydrophobic part of it, and this part what you see out here is the hydrophilic core. So, these kind of molecules, when they get inserted inside the membrane, so it is something like this. Again, I have to draw the structure, so that makes sense of the membrane. And okay. So, this is where these kind of molecules get inserted into the membrane, and here they have the potassium core through which any kind of water or any other ions can pass through, or any other molecules can pass through. This is one such example, which is carrier transport. So, there are other examples which we will be talking about it. One of them is your valinomycin. This is another cyclic molecule. It is something like this. Let me put all the residues, and then I will tell you A, B, C, D, and this A, B, C, D stands for different residues. I am going to come to that. A, B, C, D, A, B, C, D, A, B, C, D. So, sum total there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 12 such residues, where A stands for L, it is an L isomer, L lactate. B stand for L valine. C stand for dihydroxy isovalerate, dihydroxy isovalerate, and D stand for D-valine. And if you go through the structure of it, it is something like this. So, CH O minus CH3 C double bonded with O. So, this is A. So, now come to the B. B is NH CH, and you have CH, you have CH3, you have CH3 here, and you have a C double bonded with O, and this is your B. Then you have the C molecule, which is oxygen, C, CH, CH3, CH3, C double bonded with O, H, this is C, and there is another one here, NH, CH, C double bonded with O, CH, CH3, CH3. So, this is D and this is C. So, this is how it is being connected in the valinomycin, and when this valinomycin molecule, I showed you that and then A, B, C, D. Likewise, let us see A, B, C, D, A, B, C, D, and then again A, B, C, D. And within the center of it, what you observe is something very interesting. Again, there is an potassium which is present in the center and which is kind of coordination complex, and it is this potassium which is there which allows the ions to move through it. Okay. In the same line, there is another molecule. These are kind of the classic molecule which have been used all these years, is called gramicidine A. Gramicidine A is another such channel forming molecule. So, gramicidine is even much more complex structure of let me put the structure for you guys, so that you kind of get a feel of these kind of structure. These are the small structure. These are easy to understand before we understand. So, this is an alternate L and D isomers of amino acids. So, this is the L isomer, then you have glycine, then you have the alanine, which is the L isomer here. Then you have, I kindly request you people to kindly go through the structure of the amino acids from the notes. From alanine, this L, this is up to 5. Then you have the valine, which is in D conformation. Then you have another valine, which is in L conformation. And you have another valine, which is in D conformation. You have tryptophan, which is in L conformation. You have a leucine, which is in D conformation. Okay. So, you see there is an alternate L and D, and then you have tryptophan, which is in L conformation. You have leucine, which is in D conformation. You have another one which is in L conformation. You have leucine in D conformation. You have TRP in L conformation, and then it is attached to a carboxylate group, and NH, CH2, CH2, OH. So, this is how the gramicidine molecule looks like, and all these molecules have alternating L and D isomer, and they are the one which form the channels. And these are some of the probes much before the membrane structure could be derived, and it was a very challenging task. These are some of the molecular probes by which ion channel structures have been studied for several years, and now also they are being studied. These are small molecules. They act as antibiotics, they act as toxins. There are series of them. I just picked up 2 or 3 examples like valinomycin, gramicidine, and carrier antibiotics, which could help you to appreciate how the membrane must have developed. Okay. So, from here we will move on to the fourth topic of this section, which is the flow of ion through. So, I showed you that diagram in the beginning of the black lipid membrane. So, say for example, let me redraw it. So, you could recreate the membrane like this, and here you have this lipid bilayer formation, and here you have the electrodes, and you have a very high gain amplifier to record the change in voltage across it, and there are different concentration of say sodium, say may be high on this side, sodium ions may be low, chloride may be high here, or chloride may be high here, or say for example potassium may be low here, potassium may be high here, whatsoever the flow of this could be the flow of ion through single channel in a membrane can be detected. So, basically through single channel can be detected. How it is being done? This is a very straight way. So, I showed you that using a brush, let me go to the next slide. So, you use a brush to create that membrane structure. So, along with that brush you mix some of these channel forming peptides or channel forming molecules. So, what will happen is this. Now, out here you will have you have some of the channel forming structures which will form, and they will allow the flow of ions across either one of the sides, and that will help you to record the flow of ions through those channel. Okay. So, this is how most of these channels are being studied, and the recording kind of comes like this. If this is the baseline, then you will see something like. So, this is when it is going up, is basically the channel is getting open. You can measure the density of channels, you can do a whole bunch of complex experiment, and you can understand how the channel looks like, where x axis is showing you time and y axis is showing you conductance. And we will talk more about it, about the single channel recording, as we will move through, go to the ion channel, where we will be talking about some of the fundamental discoveries, ion channel recordings, which were done by Irwin Nihar, Bert Sackman, Alan Marty, and all these other people, which won them a Nobel Prize and many other things. Okay. But just for your interest, I can tell you that across these gramicidine channels and everything, almost 10 to the power 7 cations could travel per second. This is almost the rate of migration, which is a huge number. Okay. So, with this we will move on to see. So, we kind of give you an introduction about how probably even bigger structure. So, we talk about very, very small structures, okay, very, very small structures which are forming channels. Now, think of it a huge protein like structure, how it will look like. There are several conformation and geometries how it looks like. So, let us draw the structure of the membrane to tell you how the proteins look like. Okay. So, now we are entering into the area of membrane proteins, which are embedded within the lipid bilayer. Okay. So, let me again redraw the membrane. Sorry. Okay. So, the proteins I will draw it in black to give you an understanding. Some of the proteins span like this from the one side to another side, they are like this. Okay. Yet there are proteins which sits on top of it like this, okay, like this. It could be on the outer side, it could be on the inner side, in both sides. Okay. Yet some of the proteins just have something like this, one part and maybe attached to another part like this. So, this is how all these different kind of proteins are sitting all along the membrane structure, and these proteins could be your could be on the peripheral side, on the periphery, it could be integral protein, it could run all across the membrane, How these things were discovered, that is very important for you people to appreciate. So, there are certain techniques by which these have been done. There is a technique called freeze fracture etching technique. So, what do you do? Say for example, let me take some, say for example, this is the bilipid membrane. Okay. Just think of my purse as the bilipid membrane. So, let me see how I orient it. Okay. So, imagine underneath is inside the cell and up it is outside the cell. What I do, I take this stuff and I give me 1 minute. Okay. What I do, I freeze it down at a temperature of say liquid nitrogen, exceptionally cold temperature. Okay. I freeze it down. As soon as I freeze it down, this is what will happen. Let me okay. Let me the membrane. So, this is your membrane structure, what I was trying to show you. Okay. So, and here is your protein of choice. Okay. So, now I bring down the temperature to liquid nitrogen temperature. Okay. So, the temperature is of liquid nitrogen, and then what I do is at this stage I bring a sharp knife which is called a microtome knife, and I cut the membrane, split up the membrane, and this is called, basically the whole technique is called freeze etching, ETCH, freeze etching electron microscopy. This is how it was discovered that there are integral proteins which run from one side of the membrane to the other side of the membrane, electron microscopy. Okay. And what you get out of this is that doing this deep etching is something which I will show you through the purse using my wallet. Okay. So, say for example, this is the outside and this is the inside, and this is the lipid bilayer for you guys. Okay. So, when you put this at liquid nitrogen temperature and you cut it, so you take the knife, say for example, let me see. So, say for example, this is the knife, okay, and I take the knife and I cut it through like this, or I cut it through like this. What you will see is that it opens up like this, and then this is exposed in front of the microscope, in electron microscope, and there you could see if there are some proteins which are, suppose these my fingers are the proteins which are there. So, I can see the structure of the proteins. So, this is how it is being done. So, this technique is called freeze etching electron microscopy, and it follows a deep etching process. This is how most of these integral proteins have been discovered, that they run from one side, one end to the other end of it. Okay. So, this is one such technique which I wish to expose you people. So, from here we will move on to another technique. You remember in the last class we talked about that the lipids can move across the membrane laterally, and very rarely it do a flip flop movement. It is really tough for it to do so. So, how it was being proved, and I told you that I will give you some of the techniques by which membranes have been studied. So, one such technique is like this. Say for example, so let us put the title. So, this is in this section we will be talking about lipid and many membrane proteins diffuse rapidly in the plane of membrane. So, how to prove this? So, we need experimental proof to do so. So, this is how it is being done. Say for example, I have one cell A, say for example, cell A like this, and I have another cell, and let me put a different color, cell B like this. Okay. These are the two cells, and on these cells I have some kind of a tagging of the lipids and the proteins. Say for example, these are the tagged. Okay. You see these are the blue colors are the tags, and the other one I have this black tag. Okay. So, you have the blue tag, you have the black tag. The next what I do is, let me pick up another color to explain it easily. I fuse these two cells. If I fuse these two cells, so the structure will form like this. Okay. I am fusing these two cells. Once I fuse these two cells, so what will happen? This black will remain here, and all the label things will remain here. And after some time, what will you observe is this. After you just give it some time, and then you observe the new structure, which is almost this big, and if I give the real color something like this, like so let me check through. Okay, green and red. Okay, fine. Green. So, if this part is the green one and this part is the red one, what I will see is all those black dots and all those blue dots have mixed together likewise. So, that is one of the indirect proofs or kind of a crude proof to say these proteins and the lipids are moving all across the membrane. There is another way to prove it, which is much more quantitative. So, this diffusion takes place. So, there is a diffusion of lipids and proteins. Okay. There is another way to do so. That technique is much more quantitative. So, that is called fluorescence photo bleaching recovery technique. Photo bleaching recovery technique. What this technique is about? This is a very interesting technique. So, this technique is like, say for example, I have a cell like this. Okay, I have a cell like this. Fine. And in this cell, so this is the nucleus and all those things. Okay, and this is the membrane. Okay. On this cell, what I do? I tagged it with some kind of a fluorescent probe. So, these are the fluorescent probe. These are fluorescing. Okay. Fine. So, then what I do? I pick up one spot. Say for example, I pick up this spot and I put my microscope at this spot. So, here is my microscope, you could see here is my microscope, I am seeing this spot. So, what I do? At this point I record, say for example, I have x axis, y axis. So, I am recording fluorescence intensity on y axis. Okay. Fluorescence intensity on y axis and x axis is time. So, initially what I see at this spot, there is a fluorescent intensity like this. Let me put it in red, that will make more sense. Okay. So, then what I do, next thing, I bring a very strong laser beam and here is a laser beam coming, some kind of a strong beam which kind of bleach the whole surface, or in other words, bleaching the whole surface means it get rid of all the fluorophores which are present out here. So, all the fluorophores which are present out here. So, it is free from all the red. So, what you will see in the graph is that immediately the fluorescence intensity will fall down like this, sharp fall, because of this is where you are bringing the bleaching coming into play. After some time, what you see is that there is a recovery, it started pulling back. What exactly is happening? Why it recovered? It recovered because all the surrounding these molecules, what you see, these molecules started moving to the bleached site like this. In other word, there is a mobility and the movement of the different proteins and the lipids which are present. So, these are two standard techniques by which it has been proved that there is a mobility of the lipids and the proteins across the membrane, and very rarely you will see a flip flop movement, which I explained in the last class. Okay. So, membrane proteins do not rotate across the bilayer membrane bilayer, but sometime the lipids do so, but very slowly. Based on all these, we moved on to the. So, there are over the last century several models of the membrane has been proposed. As we are advancing, as we started understanding, many of the older models got discarded, and the most current model, which is fairly acceptable, is called fluid mosaic model. This was proposed by in 1972 by Jonathan Singer and Garth Nicholson. Okay. So, fluid mosaic. You understand mosaic? Mosaic is just like in your house, you have the mosaic floor. So, it is kind of mosaic like. This is a mosaic of proteins in the lipid. Okay? Something like that, how you can in 1972 by Jonathan Sanger and Garth Nicholson. These are the two people who proposed that. Nicholson. What they proposed is, that was their basic definition on it, is the membrane are two-dimensional solution of oriented lipids and globular proteins. So, this is their central theme about the model, and which consists of the lipids like phospholipids, glycolipids, which act as a solvent to embed the proteins in them, and there is a lateral diffusion which we have already discussed. Okay. From here, we will move on to the next topic, which is the asymmetric nature of the membrane. We have already discussed this, we will not discuss much on it, except the fact that, as I have mentioned, the proteins never flip flop, and that is what helps them to maintain the asymmetric nature of the membrane. Okay. So, it is something like that. There are certain pumps called, we have not talked, one of the major pump is called sodium potassium ATPase pump. So, this pump has the ability to throw sodium outside the cell and takes potassium inside the cell, and this only does when ATP is present inside the cell. Always remember, ATP has to be present inside the cell. Okay? And this is the membrane which I draw. This is the membrane and this is the pump. Okay? And this offers an asymmetry. This offers an asymmetry in terms of the ion concentration inside and outside the cell. Okay? So, there are certain toxins which binds from outside, which Oben is one of them, which binds outside and block this pumps, and these pumps and these channels, they offer a lot of asymmetric nature to it. So, now what controls membrane fluidity? Membrane fluidity is controlled by the fatty acids composition and cholesterol. How it does so? So, say for example, if the fatty acids are arranged like this, if they have a all straight chain like this, the hydrophobic tail is very straight, and that is only possible when the hydrophobic tail is in trans configuration. Okay. So, that situation it will be a very compact structure, whereas think of it, if the hydrophobic tail is something like this, where there is a lot of cis configuration coming into play. So, this will have this will be not very compact, this is much more disturbed or much more random structure. Okay. These structures have low melting point, Tm, which represent the melting point, they will have a low melting point. So, in other words, and whereas as compared to a very rigid structure. So, there are three parameters which decides the length of the fatty acids, the kind of bonding they have, saturated or unsaturated, saturated or unsaturated. In other words, the presence of single or double bond, and on top of that the presence of cholesterol, where we started this class. The cholesterol decides a lot about how the membrane fluidity is going to change. The last 1 minute I am going to devote now about the carbohydrate. So, if this is the membrane, most of the carbohydrate points on the outer side. If this is O is out and I is in, the carbohydrate moieties are outside, either attached on top of proteins like this, if this red thing is the protein, or they are directly attached to the lipids. Okay. They could be a glycolipids or they could be a glycoproteins. Okay. And apart from it, there is another technique, last technique, which I just want for your information. These membrane proteins could be isolated using several detergents like Triton, DDAO, they have several full forms, that is dodecyl dimethylamine oxide, sodium cholate, and octyl beta glucoside. These are the different detergents which could be used to isolate the membrane proteins. So, I believe this is what is essential for you people to understand, that membrane is a very dynamic structure, it is asymmetric in nature, it is extremely fluidic, and all these parameters changes whenever there are some changes in the body. And with this we will move on to the, we are closing on this lecture, and in the next lecture we will talk about the membrane of the nerve and muscle. Thank you.